Okay, so we're going to talk about how you screen a genetic library. It can either be a cDNA library or a genomic library. Okay, and what we've got here is on the left, this is one kind of library where you have bits of your clone DNA which are drawn in yellow. So that's, let's say it's human cDNA or human genomic DNA, and you've got loads of plasmids. Each plasmid, loads of cultures of E. coli. Each culture of E. coli has a slightly different plasmid in it, because the plasmid's got a different bit of clone DNA in there. Okay, or you could have the clone DNA rather than being in a plasmid vector, you could have it in a bacteriophage here. So these are viruses which infect E. coli. You've got the little bit, the yellow bit here is the bits of human DNA, which is sandwiched between two bits of viral DNA. And you've got loads of those clones. And each one has got a slightly different bit of human DNA in it. Now, let's say you are interested in looking for a particular gene. So you might be looking for insulin, or it could be the alpha chain of hemoglobin, I could use that example, or the beta chain, or it could be a cytokine like interferon or interleukin. It could be any protein. Let's say it's the alpha chain of the hemoglobin. So you want to find out that. So you want to find out which clone has got a piece of DNA for that. So what you need is you need a partial sequence here. You've got a sequence there. Now you'd actually want it to be slightly long. You probably want it to be about 20 long, 20 base pairs long. I'm not drawing all of them. And you make a radioactive probe. So it's a radioactive piece of DNA with a complementary sequence to that. So if it's got a complementary sequence, it will, so that's G, T, A, C, so on. Yeah, that will hydrogen bond to the other strand, sometimes called hybridizing, or sometimes called annealing. Okay, so the bit of radioactive probe will stick to the DNA in the, you've got a Petri dish here with all your clones there. So you've got lots of different clones containing different plasmids or different viruses. And to one of them, when you put your photographic plate over it afterwards, one of them, or two of them maybe, will give you a dark spot on the photograph, which means that you've got the radioactive probe hybridizing to the DNA. So that means you do have a bit, in this case, you have a bit of alpha globin DNA in that clone. That's the way of doing it. So that all sounds easy, but then you've got to think about, well, how do we know what this sequence is? How do we know what it is? Because these experiments were mostly done 20 years ago, before the Human Genome Project, so didn't know the sequence of every gene, and people use these techniques to work out the sequence of lots and lots of different genes before the Human Genome Project. So how do you know that? Well, one way of it was to get if you've got a partial protein, partial amino acid sequence, okay? So you don't have to have the whole protein, which may be 300 long. You'd only need to have it to be quite short. 7 amino acids is enough. 7. Now, 7 amino acids, from that you can infer the nucleotide sequence, the base sequence. So three bases for each amino acid, so that's going to be 3 times 7. That's going to be 21 long. So you're going to do a sequence of 21 bases long. Now, just to put that into perspective, the chances of finding that randomly, that sequence just randomly, would be, there's 4 bases, so 4 to the power of 21, which is bigger than 10 to the 12, okay? So the chances of finding that randomly would be 1 in 10 to the 12. There are only 10 to the 9, 3 times 10 to the 9 base pairs in the human genome, so you would not find that randomly. It would only occur once. So that's plenty long enough. It doesn't have to be the whole gene. But there's a problem there, because if you remember that the genetic code is degenerate. So what does that mean? Well, let's imagine if we have, I'm not going to do 7 amino acids here, but let's just do 2. Let's do glycine. So we've got glycine and we've got alanine. Okay, so there's two there. Now, if you look at glycine, the codon for glycine, the code is G in the mRNA is GG. So the codon is the mRNA. GGU, remember U replaces T in mRNA. But the last base, although all glycines are all G and G, the last one can either be U, C, A, or G. So there's 4 different possibilities there. Alanine, well, that's UC, and then once again, the last base can be variable. It can be U, C, A, or G. So with just 2 amino acids, you can see there, you would have to make, there's a total of difference of 2 amino acids. There are 8 possible base sequences. So you're going to have to make, so if you've got like 7 amino acids, you can see there's a lot of different possible base sequences. And you're going to have to make each oligonucleotide. Remember, an oligonucleotide is a bit of DNA with a few bases in it, so 21 bases in this case. You're going to have to make all of them because you don't know which one is going to work, and that's expensive and time consuming, and so it's not a brilliant way of doing it. Incidentally, you would probably also want to avoid amino acids like serine, because serine can actually have 6 different codons. And it's not just the last base that varies with serine, the kind of don't seem to be particularly related at all. It's just lots of different ones, 6 different ones. So a way around that, well, more recently, there's something that can be used in the last 10, 15 years or so, something called an expressed sequence tag. Or an EST. Now, what that is over the last 20 years or so, whenever people make a cDNA library and sequence it, they publish the sequence of it, and it goes into a public database, and it's stored there. And there are like literally millions of cDNA sequences from humans in the database, and no one knows really what they do. If it's a cDNA, you know it must have come from RNA, so you know they refer to coding sequences of proteins, but no one's actually that sure what they do. So what the approach is to get your get these oligonucleotides. So you want to get every possible oligonucleotide, every possible base sequence for the 7 amino acid stretch. Okay, so that's taken into account that the different, most amino acids have 4 different codons. So you make every possible one, and you check for a match. And then you probably will find one match, at least one match. So, and as we said, that must refer to a protein, to an actual protein sequence, because the chance of it just coming up randomly is like 1 in 10 to the power of 12. But if you've got 20 nucleotides, so this is not going to happen randomly. So it must refer to a protein sequence, and it probably is the protein that you're looking for. So then the approach is to use this sequence as the probe. So that means you only have to make one probe instead of making all the different possibilities. So that is using this thing, an express sequence tag, millions of cDNA sequences in a database, and they'll really know what they do. Now, the other approach is not to use a DNA approach at all, DNA sequence at all, is to look at the protein which is made from the gene. Okay, so you're going to look at the protein made from the gene you're interested in. Now we keep saying alpha globin, yeah. The alpha globin, the alpha chain of hemoglobin, alpha globin. So what you need is you need some monoclonal antibodies which recognize alpha globin. Now, the kind of library you need to use here is a special library called an expression library. Expression means that the bit of DNA doesn't just sit there and get replicated with a plasmid very often. So there's your plasmid. Yeah, it gets replicated in, and you get lots of copies of your inserted yellow bit human there, but it doesn't necessarily get turned into mRNA and translated and turned into protein, because that just doesn't work. So you need to use an expression vector. Sorry, and plasmids are not that good as expression vectors. They use a special kind of phage lambda, a special manipulated phage lambda called phage lambda GT11. I don't know why it's called that, but it's one that's been messed about with. Now, a phage normally, this is the bacteriophage DNA, and you put in your bit of human DNA in like that. Well, then that's the normal phage lambda. In the lambda GT11, you have your phage lambda DNA. Then you have this green gene, which is called beta-galactosidase. It doesn't really matter what it's called. That is a gene which is from E. coli. And that gene can be if it's in E. coli, and it's only expressed when they're fed lactose. So the gene beta-galactosidase actually it breaks down lactose into glucose and galactose. E. coli don't make it all the time. There's no point making it, but they can respond to their environment. So there's lactose in their environment. That gene will get switched on. We won't worry about the mechanism of that, but it is switched on. This gene is switched on. So that means it will be transcribed, translated, transcribed, and then translated. As long as you've got lactose in the culture medium. And then next to that, you stick next to that, that's your bit of human DNA. Okay, it's all packaged up into the phage head as usual. So you've got your beta-galactosidase gene and your bits of cloned DNA in there. You've got a whole library of them like that. So when they infect an E. coli, that E. coli, so the E. coli will make loads and loads of those bits of DNA prior to them being packaged. So I'll just draw one or two of them. But because it's got that green gene there, the E. coli gene, and if you feed it lactose, it will make that gene prescribed, so they'll make protein. They make something called a fusion protein. So it's not the human protein, proper human protein, because it's got that bacterial gene first. So I'll tell you what, I'll do a little color code. We'll say that the protein which comes from the bacterial gene bit is blue, and the bit which comes from the human bit is purple. Okay, so the fusion protein will look something like this, it's folded, and then you'll get, so that bit is come from the bacteria, and that bit is human. That's the fusion protein, is a mixture of the two things. But usually it's sufficiently similar in shape that when you put antibodies to it, the antibodies can actually bind to that protein. So this is the monoclonal antibody to the alpha hemoglobin, to the alpha globin gene. So similar process, similar procedure to the others. What is done is the Petri dish, lots of different clones on it. That is transferred onto nitrocellulose. You get the protein transferred to nitrocellulose. You add the monoclonal antibody, and then usually a second antibody is added, and the second antibody has got the fluorescent tag on it. So very like an ELISA testing. So the second antibody is going to recognize that end of the first antibody, and so put all those both sets of antibodies on the monoclonal first, then the second one, wash off any unbound stuff, shine a fluorescent UV light on it, and if there's any antibody bound, it will fluoresce. You'll get a spot there. So you can see that that particular colony is producing the fusion protein, which of the alpha globin. So we know that that one there, the insert in this one, has got the alpha globin gene in there. Then you can get your bit of DNA with all your bacterial DNA and your actual DNA there. Get restriction enzymes, grow up loads and loads of that culture, so you've got tons and tons of the DNA, and then chop out the bit of DNA that you're interested in, and then that can be sequenced or whatever.